All right, hello to those of you who are um, joining in on this lecture. This lecture series will cover systematics as well as the concept of species and how to identify or determine species and the different mechanisms for such. To start, our living world is more or less comprised of distinct entities, which we call species. Um, this is Latin for kinds. Now, the ease at which we are able to identify species and delineate between species is variable. It ranges from relatively simple to virtually impossible. And this dilemma is often referred to as the species problem, or the kind of the Gordian knot of systematics. It's simply the inability of biologists to arrive at a universal definition for the term species, as well as a general method of discovering and delineating species. In part, this problem kind of arises from the diversity of approaches and methods that can be employed by different sectors of the realm of biology as far as identifying and distinguishing species. Because biology is such a vast realm and there are so many kind of sub-study areas of biology, it's hard to define and recognize species in a uniform manner that can be uniform or general across all those disciplines. So why is it important to be able to define and discover species? Kind of why is it important to work around and untie this Gordian knot? Well, in a sense, as it's been said before, species are sometimes considered currency of biology. And the reason for such is that the notion of species is kind of what enables biologists in different fields to communicate with one another and compare their results in a meaningful way. It essentially serves as these small building blocks and also enables us to, in a way, compare apples to apples. Another reason why we want to untie this knot is kind of to simply satisfy this ingrained tendency that we as humans have, which is to continuously ask why things happen and want this kind of insatisfiable need to know what things are. So it works to satisfy the human urge in asking what kind of animal is this, or what kind of plant is that, or why is it here, how did it get here, etc. In general, kind of the superficial definition of what a species is, a species is a group of organisms that can interbreed and produce fertile offspring. Now, again, that's relatively superficial because there are so many nuances as far as determining what a species is or kind of delineating the boundaries of species. So other kind of concepts that need to be factored in when determining whether individuals within a population are one or two different species. You have to factor in whether or not they have common descent. Species need to arise from a common ancestor, but they need to have the presence of certain morphological or genetic differences that make them irrefutably different. So kind of stemming into this or going along that notion, they need to have evolutionary duration, right? These species need to have been separated at one point in evolutionary history and have been divided and existed for a certain amount of time, relatively over a large period of time, to enable these differences to appear that really kind of separate the two from one another and allow for the separation into individual species. They must be able to have these reproductive communities wherein they can interbreed with one another, but that reproductive community excludes other species. So it's more of this species exclusive community as far as reproduction, and it's isolated from other species. And within that, you need to have an established geographic range, kind of understanding where these species occur or where they would not occur. And they have to be irreducible, meaning the smallest distinct grouping. And we'll get more into what that means when we talk about phylogenetics. Another reason, kind of going back to why it's so difficult to identify species and delineate between species, is the fact that we are continuously discovering new species. We have only identified maybe 1% of species, at least 1% of species in the Amazon rainforest. And we are still continuously finding new species, some that are existent today, some that are now extinct. So this skeleton here, these are the fossilized remains of a new species that was found recently, of an extant species called Wakaleo shooteni. This was a marsupial lion that kind of roamed in the late Oligocene, and it was discovered in Queensland, Australia. So during the Oligocene, this guy kind of roamed around the open forest habitats, and based on its skeletal structure, it's proposed that this was a predator species, but it spent most of its time in trees. It weighed about 50 pounds and was more or less the size of a Siberian husky dog. Now, its teeth suggest that it was not completely reliant on meat, but rather it was an omnivore. So this is just a cool example of a new species that we have just found. Though it's not alive today, still it supports this concept that we are continuously finding new species and making adjustments in existing phylogenetic trees and modifications to our understanding of modern day species as well as historic populations. All right, so as I said, identifying species and kind of distinguishing between boundaries or distinguishing species boundaries and natural populations can be very difficult sometimes. And there's a number of measures that are employed to determine whether populations are members of the same species or whether they represent different species. And we call these methods or recognize them as different species concepts per se. The major species concepts that are employed or recognized are the evolutionary species concept, phenetic species concept, biological species concept, ecological species concept, and the phylogenetic species concept. So we're going to go through each of those separately. Each one has its own unique way of defining what a species is, has its pros and cons, and kind of an alternative mechanism, per se, in species determination or distinguishing. All right, so we'll start with the evolutionary species concept. This is kind of the famous definition or kind of underlying understanding of what the evolutionary species concept is. According to E.O. Wiley and George Gaylord Simpson, they kind of created this concept back in the late 1980s. According to this concept, or according to their statement, an evolutionary species is a single lineage of ancestor-descendant populations which maintains its identity from other such lineages, and which has its own evolutionary tendencies and historical fate. So kind of, in other words, the underlying notion of this concept is that a species is a group of organisms that forms a lineage that has a distinct evolutionary fate from that of other lineages. They also have an origin. That origin is formed by a process called cladogenesis, which is the splitting of lineages. And they also undergo evolution. They undergo evolution by anagenesis, which is referring to the change within lineages. Also, according to this concept, a species is a group of populations that have a shared past and will have a shared future in terms of their location on a phylogenetic tree and their creation of a phylogenetic tree. So the major takeaway with the evolutionary species concept is that they are distinct lineages that have their own origin and their own evolutionary fate. Moving into a different concept, the phenetic species concept. This is also referred to as the morphological species concept. According to this concept, a species is defined as a set of organisms that look similar to each other and distinct from other sets. And it essentially draws clusters around groups of individuals in what you can call morphospace. So it draws clusters around phenotypically similar individuals or populations. And it uses that clustering method in this morphospace or phenotypic space to draw species boundaries between those clusters. So here you have the clustering of, or 3 clusters formed, and based on those clusters, you can create 3 separate species. These vertical and horizontal axes of the phenotype space each represent a single or multiple phenotypes or phenotypic traits. Again, those are the traits that are used to determine kind of the pattern of clustering that is created, and then that pattern is used to assign those species boundaries. Now, this works for sexual, asexual, and extinct species, which is a positive component of this concept. Now, in general, the phenetic species concept, it's more of a quantitative approach per se. It seeks to classify organisms on the basis of their overall similarity according to numerical taxonomy. Numerical taxonomy measures and records similarities for large numbers of characters. So with this concept and that quantitative approach, species, or in order to be identified as species, they contain some exact degree of phenotypic similarity. And that value of similarity would be measured by some sort of phenetic distance statistic. So essentially, a species would be a set of organisms that could be no more than X phenetic distance units apart. If they were closer than that, you would not have the separate. You would have only one species. They wouldn't be separate species, or you could have start going into more of the subspecies. Subspecies are kind of geographical grouping of species or of similar individuals. So that's kind of looking at the phenetic species concept in that quantitative approach, or what I mean by when I say that it's more of this quantitative approach. So as I said before, this concept was beneficial because it works for sexual, asexual, and extinct species, but it does have its issues. First is kind of this question of how do we weigh the relative importance of characters or traits that are used to delineate these species boundaries. Are they all weighted the same? Meaning, should they all be equally important, or should different traits be weighted more heavily? And how do we determine which ones should be weighted more heavily? Assumingly, we would weight those traits that are more important to the species' endurance and survivability as being more important, but it overlooks factors that may be equally as important that are more subtle, per se. So, how do we determine the weight or the relative importance of the characters and the phenotypic traits that are used? The second issue involves convergent evolution. So with convergent evolution, different species have similar traits, but they don't have similar traits because of common ancestry. They have these are what are called analogous structures. They are structures that are very similar, but evolved independently, and the species were able to evolve them independently because they occupied similar environments or faced similar selective pressures. So more and more work kind of in this concept realm has started to use or focus more on employing genetic information rather than anatomical or morphological data. And by doing so, you can kind of mitigate this convergent evolution problem. Again, some species will have similar traits, but they have them not because of common ancestry. So that by using genetic information, you can actually make better determinations of whether or not these are related species and whether or not they descended from a common ancestor. The third issue deals with something that's called cryptic species. So what is a cryptic species? Well, it's kind of just as the name alludes. Sometimes speciation events can happen without changes in morphology or other traits. And when speciation occurs by such, you have the creation of what are known as cryptic species. So cryptic speciation is a biological process that results in a group of species, which by definition cannot interbreed, that contain individuals that are morphologically identical to each other, but belong to different species. So morphologically, they're identical. Genetically, they are not. An example of this we'll talk about later in the lecture are your poison dart frogs, but also Arctic warblers. Arctic warblers are all very similar in appearance, but they occupy different geographical areas, and they have stark differences in their genetics that show they actually belong to different species. So each one of these represents a different species. Not all of them are the same species. All right, so moving on to the third concept. We'll talk about the biological species concept. This concept was first introduced by Ernst Mayr. And according to this concept, a species is composed of groups of actually or potentially interbreeding populations, which are reproductively isolated from other such groups. So here species boundaries are determined by the patterns of gene flow. In other words, is gene flow present or absent among populations? And based on such, you can delineate these species boundaries. So it's not so much based on the attributes of individuals per se, but rather it operates by delineating species according to properties that are possessed by populations. So it's more of a population view rather than an individual view. Again, this method has its issues and its limitations, so it's difficult to apply this concept to extinct species. That's one major issue. Another is that it's restricted to sexual species. In asexual reproduction, each individual and every clone is technically reproductively isolated, according to Ernst Mayr. But we cannot go about naming each one as a new species. That's just exotic, and we physically don't have enough materials to go about doing that. It would be such a large endeavor. The third limitation is that kind of dealing with hybridization. So hybridization events between individuals and populations are technically reproductively isolated. So do we determine all of these kind of hybrid offspring as new populations, or how rare do these cross-population matings have to be before that we can say that the two populations are truly two separate species? Also, how poor must the hybrid offspring fair or exist before we can make that determination. So those are the three major issues. Again, difficulty to apply to extinct species. It's only restricted to sexual species. And how do you deal with hybridization and account for that in determination of species? So kind of an example of this concept would be your meadowlarks. There's the western meadowlark and eastern meadowlark. They both appear to be identical. They have very similar coloration patterns, as well as size and morphological features. They also have overlapping home ranges, but they do not recognize one another, or they do not interbreed with one another because they don't recognize the courtship rituals or reproductive strategies of the other species. So they're distinct. They have distinctly different songs and distinctly different courtship strategies that prevent interbreeding. So this is also an example of biological isolation. Interbreeding between these two species is prevented based on behavioral preferences. In regards to this case, the preferences are for individuals of that species. All right, so moving on to another concept. I think this is our fourth concept, is the ecological species concept. The ecological species concept defines a species as a set of organisms that exploit a single niche or a given set of niches. You can also refer to niches as these adaptive zones. It kind of uses the scope of ecological competition to delineate between species and species boundaries. So two lineages have evolved adaptations over time that enable them to occupy different niches, then they are designated as different species. Individuals that are said to be members of the same species are usually individuals or populations that compete closely or whose descendants can potentially replace one another. So you have a lot of niche overlap with those. Again, those would be considered the same species. So the integrity of a species, or what determines a species, is not so much this notion of reproductive isolation, which is kind of talked about in some of the other concepts as far as recognizing gene flow, or that you, with like the biological species concept, there are these interbreeding populations which are reproductively isolated from other groups. This concept's not like that. In this concept, species integrity is more determined by each species' ability to adapt to a niche and therefore exploit and occupy different niches. So pros and cons to this concept. This is a good concept in that, unlike the biological species concept, it doesn't have limitations based on hybridization. So here, hybridization is not a problem, particularly if hybrids are less fit or have restricted ranges. This concept also works for asexual species, as well as sexual, reproductive. The problem with this concept, though, is that there's difficulty in defining niches independently, and more so determining niches kind of independent of the species that occupy them. So how do we determine these niches, differentiate them, and determine whether or not they truly are different niches? Some, again, it's almost like the species problem. Some are relatively simple, others not so much. Alrighty, so on to the last species concept. This is the phylogenetic species concept. So like the phenetic species concept, the phylogenetic species concept kind of looks to distinguish species based on character differences, but it does so in a different fashion. According to this concept, we draw species boundaries using shared derived characters. And these characters are unique to one monophyletic group and absent from all other populations when they're plotted on a phylogenetic tree. And we call these characters synapomorphies. And those synapomorphies, again, would be used to distinguish among species, wherein each species represents a monophyletic group. Right? This is what I was referring to when I said one of the kind of ways to define a species is that species must be the smallest comprising unit. They must be irreducible. This is what I mean by that. A species on a phylogenetic tree must be the smallest monophyletic group. Again, each group is distinguished by a shared derived character. So any population that forms an independent branch on the phylogeny, as you can see here and here, would be recognized as a species or species grouping, depending on where you're actually drawing those boundaries. So again, it uses synapomorphies which are your shared derived traits, to determine and distinguish species and species boundaries, more or less between monophyletic groups. Each branch tips represent populations or different species, and usually on these trees there's these kind of colored markers. These tick lines represent a trait that serves as a shared derived character. So using that, you can kind of diagnose where these species originated and what derived trait categorizes them as separate species. So kind of another important point as far as this concept, there's actually two other points I want to mention. In order to be called a separate species under this concept, populations must have been evolutionarily independent for a long enough time in order for these diagnostic traits to emerge and differentiate. Right? So that goes back to that notion in kind of ways that we define species as far as having evolutionary duration. There's also kind of restriction onto the types of characters that can be used for determining or delineating these monophyletic groups or your species. The phylogenetic species concept can't use polymorphic characters. That's because they are not shared derived characters of a monophyletic group. For example, talking with flowers, flower shape, its unique shape and pattern is a shared derived trait of a monophyletic clade. So that can be used to delineate species boundaries. But a character state like flower color, on the other hand, that is highly polymorphic. There's a lot of variations that you can see in color within a single species. So that's, and it's also not a shared derived trait. So that can't be used as kind of a synapomorphy for distinguishing species and creating those monophyletic clades. All right, so those are your species concepts. Oh, you know what? Actually, before I move on, so this is a very interesting example of some kind of novel research that has just been done recently, kind of showing the importance of using phylogenetics and kind of the work of this phylogenetic species concept. For a long time, it was considered that you had Asian elephants and species of African elephants, but it wasn't really considered that there was a great deal of species differentiation between these two types of African elephants. There's your African forest elephants, which are more from the tropical forests of Africa. And the second type are the African savanna elephants, or your bush elephants. So they have some morphological differences, but it wasn't, for a while, it wasn't recognized that these were two majorly separate species. Several years or so ago, DART biopsy samples were gathered from both of these populations, and they underwent phylogenetic analysis, looking more at the genetic differences between these two genera. And they found that there was an enormous amount of genetic difference and distance between these two populations. There were multiple genetically fixed nucleotide site differences. There were, again, also these morphological and habitat distinctions, as well as this limited hybridization of gene flow between the two populations. So based on all of that information, particularly the new information that was gained looking at the genetics and comparing the genetic sequences of these two populations, it's determined that these are two separate species. So the African savanna elephant is a separate species from the African forest elephant. The two species actually appeared to have evolved from a common ancestor between 2.5 million and 5 million years ago, which is nearly as long as the human and chimpanzee lineages, or when that divergence occurred. So what they found also, the differences, the genetic differences between the two are so apparent. They are just about as genetically different as your Asian elephants are from woolly mammoths. So again, incredible kind of use of different character states to determine the phylogeny of species and determine whether or not these are different species. And when we talk about or go more into phylogenetics, you'll come to recognize that multiple factors or types of characters are used to build phylogenetic trees. You can't just use morphological traits or genetic traits. You have to use compilation of a variety of different character states to create more evolutionarily accurate delineations and depictions of species and their evolutionary history. So that's kind of the bulk of what species are and the different methods that are used for determining species as far as the different species concepts. The lecture following this will delve more into this road of systematics by going into phylogenetics and phylogenetic systematics. But I hope that this lecture helped you understand different ways that species are determined, and again, just how complicated it is to determine whether individuals are or are not different species.